Hello all, this is Chapter 24, Chromosomal Basis of Inheritance. So we're going to talk about gene linkage. So a single chromosome contains many alleles or genes in a definite fixed order. And it's fixed because each allele has its own location called a locus. And a linkage group are all the genes on one chromosome that tend to be inherited together. So the closer they are, the more tightly they will be linked. Two trait crosses assume the genes are on non-homologous chromosomes. Genes that are linked do not show independent assortment. So here are different loci on human chromosome 19, different genes. So you can see we have the muscular dystrophy gene, eye color for green-blue, hair color, deafness, a gene that's linked to ovarian carcinoma, and other genes as well. So genes A and B are located close together and are inherited together. Recombination is rare. Only two types of gametes are produced even though chromosomes are dihybrid. Genes A and B are farther apart and crossing over can occur between them during meiosis, and four types of gametes are produced. So during meiosis, crossing over sometimes occurs between non-sister chromatids in a tetrad. And chromatids can exchange genetic material. If crossing over occurs, a dihybrid produces four types of gametes instead of two. The occurrence of crossing over can help tell the sequence of genes on a chromosome, because crossing over occurs more often between distant genes than between closer genes. So normally both male and female humans have 23 pairs of chromosomes. 22 pairs are called autosomes, and one pair is the sex chromosomes, and they differ between the sexes. Human males are XY and human females are XX. The X and Y are non-homologous and contain different combinations of genes. Sex-linked traits are controlled by genes on the sex chromosomes. X-linked genes are found on the X chromosome. There's roughly 2,000 genes on this chromosome, and no matching genes on the Y. And there are some Y-linked genes found on the Y chromosome, and there's roughly 200 genes on the Y. For X-linked traits, a male always receives an X-linked allele from his mother from her X chromosome. The Y chromosome from the father does not carry an allele for that trait. Most X-linked genetic disorders are recessive, and the females must receive two recessive alleles, and it's one from each parent. When examining X-linked traits, the allele on the X chromosome is shown as a letter attached to the X chromosome. The example here is red-green of color blindness. So X with a large B is normal vision, and X with a small b indicates of color blindness. And this is a well-known X-linked recessive disorder. And females can be carriers, which means they have one mutant allele, but they still do not have color blindness. So there are more colorblind males than females. So here's a cross with X-linked genes for color blindness. We have a male with normal vision and a heterozygous female. So what is the chance they will have a colorblind daughter? All daughters have normal vision because they're the XB from their father. What is the chance they will have a colorblind son? Sons have a 50% chance of being colorblind because they can either inherit the normal X chromosome or the mutant X chromosome or disease X chromosome from their mother. And here's a mating setup in a Punnett square. We have a key and a phenotypic ratio. So you can see here, only a male can become colorblind from this mating, and it's a 1 in 2 chance. So most sex-linked disorders are carried on the X chromosome. More males than females will have the disorder. The recessive allele on the X chromosome is always expressed in males. because the Y chromosome lacks an allele for the disorder, and disorders are often passed from grandfather to grandson. So here's an X-linked recessive pedigree you can take a look at. All right, remember all the yellow people are affected, and there are the rules listed out below. So more males than females are affected. A son who is affected can have parents who have the normal phenotype. Excuse me. For a female to have the characteristic, her father must also have it, and her mother must have it or be a carrier. Often skips a generation from grandfather to grandson. If the woman has a characteristic, all her sons will have it. X-linked dominant disorders, only a few are known. Affected males pass the trait only to daughters who have a 100% chance of inheriting the disorder. Females can pass the dominant allele to both sons and daughters, causing the disorder. And an example of this disorder is incontinentia pigmenti, and it causes discoloration of the skin. So here are some of X-linked recessive disorders of interest. Color blindness, so it affects 7 to 10% of Caucasian males. And bright greens are seen as tans and reds as reddish brown. There's Duchenne's muscular dystrophy, which is characterized by a wasting away of the muscles. Symptoms include a waddling gait like a duck, toe walking, and frequent falls. And it's caused by the absence of a protein called dystrophin. Here's some X-linked recessive disorders. So there's Fragile X syndrome. It's caused by an abnormal number of repeat sequences in the genome on the X chromosome, and it's the most common cause of inherited mental impairment or retardation. And there's a wide range of phenotypes here, from very mildly affected to very severely mentally retarded. Males also have characteristic physical abnormalities, and it's less frequent and milder in symptoms in females. Hemophilia, it's a disease caused by the absence or minimal presence of a clotting factor. Two types. So hemophilia A is caused by a lack of clotting factor 8, and hemophilia B is caused by a lack of clotting factor 9. The affected individual's blood either doesn't clot or clots very slowly, so they bleed excessively after injury or surgery. And here's a pedigree showing X-linked inheritance of hemophilia in European families, royal families, passed down through the generations. So there can also be changes in chromosome number. Except for red blood cells, any cell can be a source of chromosomes for examination. And cells are stimulated to divide with a chemical, then used to stop cell division when chromosomes are most compacted. And stains are applied and are photographed. And when the chromosomes are arranged by pairs according to their size and general appearance, this is called a karyotype. Here's a picture of a karyotype of a human here. And this is a male. You can see the 22 autosome pairs, and then the X and Y, the sex chromosomes at the bottom right. Humans normally receive 22 autosome pairs and have 2 sex chromosomes. However, some individuals are born with either too many or too few autosomes or sex chromosomes, and this is most likely due to non-disjunction in meiosis, or the failure of chromosomes or sister chromatids to separate during meiosis. So non-disjunction can occur during meiosis 1, or it can take place during meiosis 2. So the resulting gametes have incorrect numbers of chromosomes. This results in trisomy having 3 chromosomes, or monosomy having 1 chromosome when fertilized. Here is the diagram showing how non-disjunction can occur during oogenesis. So you can have non-disjunction in meiosis 1 or meiosis 2 before fertilization. So normal development depends on exactly 2 of each kind of chromosome. Trisomies are tolerated better than monosomies. And only trisomy 21 Down syndrome has a reasonable chance of survival after birth. Most of these fetuses that have changes in chromosome numbers are spontaneously aborted. And trisomy 13 and 18 also occur, but individuals rarely survive past the first few months of life. So here are the syndromes and changes in chromosome number. So Down syndrome is when you have 3 chromosome 21s. It's the most common autosomal trisomy among humans, and it has easily recognized physical features. And chances of having a Down syndrome child increase rapidly with age, starting about age 40. And the extra chromosome can occur in egg or sperm. And some symptoms may be due to the extra copy of the GART gene on the long arm of chromosome 21, which causes high levels of purines in the blood and mild to severe mental impairment. And this diagnosis is possible via karyotyping. So you can see the karyotype here of a person with trisomy 21. Circled in the karyotype is they have 3 chromosome 21s instead of 2. You can also have changes in the sex chromosome number, all right? Just like with the autosomes, it happens during meiosis. And this can lead to various syndromes. So Turner syndrome is when a female lacks an X. We have Jacob syndrome where there's an extra Y. Klinefelter where the male has an extra X. And you also have the poly-X syndrome. So yeah, a person with only 1 X chromosome is female in Turner. Person that has more than 1 X chromosome plus a Y is male, and this is known as Klinefelter syndrome. And the presence of Y chromosome, not the number of X, determines maleness. So there's a gene on the Y chromosome called SRY or SRY, and it produces a hormone that creates the testes. So deletion of this SRY gene on the short arm of the Y results in Swyer syndrome or an XY female. And these individuals lack the testes determination factor. And movement of the SRY gene on the X chromosome results in De La Chapelle syndrome or an XX male. Men will exhibit undersized testes, sterility, and they'll have rudimentary breast formation like a female. All right, normal XX females have 1 X chromosome inactive. In a zygote with 1 X chromosome, Turner syndrome can survive. All extra X chromosomes become single, 1 becomes deactivated, and these are poly-X females and XXY males are seen frequently. Extra Ys are also tolerated. And it says chances of survival are greater when trisomy or monosomy involves the sex chromosomes. All right, in normal XX females, 1 of the X chromosomes is inactivated and becomes a darkly stained mass called the bar body, and it's inactivated, therefore none of the genes are expressed. Therefore, cells of females function with a single X chromosome like males. Turner syndrome of females have 1 X chromosome. They're usually short with a broad chest, and most have ovarian failure, do not undergo puberty or menstruate without hormonal therapy, and their breasts do not develop. Klinefelter syndrome is a male with 2 X's and 1 Y. Their testes and prostate gland are underdeveloped. They usually don't have facial hair. They have some breast development and have large hands and feet and very long arms and legs. Usually slow to learn, but do not have an intellectual disability unless they have more than 2 X chromosomes. And they require assisted reproduction if they want to father children. Poly-X females have more than 2 chromosomes and thus extra bar bodies. There's no distinctive phenotype except usually taller and thinner. Some have delayed motor and language development, and some have menstrual difficulties, but most are fine and are fertile. 4 X chromosomes are more rare, usually tall, tend to have severe intellectual disabilities, and various physical abnormalities are seen, but they menstruate normally. We have Jacob syndrome, where it's an X with two Ys. These males are usually taller, have persistent acne, and tend to experience speech and reading problems, but they are fertile and may have children. There's typically no behavioral differences between an XY and XYY male.